Hi everyone. I've been working on risks associated with organic soil amendments. So today I will focus on risks related to microbial contamination in compost. Employment of composting guidelines have been put in place to reduce the risk of microbial contamination of fresh produce. Aerobic composting are expected to achieve a minimum temperature of 55 Celsius degrees for at least 3 consecutive days, according to Australian standards. However, a survey tested 103 compost samples has found 30% of samples was positive for E. coli, with level far exceeding microbial standards and even reaching 1 million cells per gram. Induction into viable but non-controllable cells, VBNC, has been viewed as one of the microbial stress responses during heat treatment. The virulence of VBNC's pathogens has been proven to be recovered after resuscitation, and resuscitated VBNC pathogens may be involved in several foodborne outbreaks. There is lack of research considering treatment efficiency of composting technology against inactivation of bacteria having the potential to enter to VBNC state during composting. Thus, the aim of this study is to investigate whether E. coli could enter into VBNC state and withstand the thermal temperatures associated with composting. In my study, I investigated the effect of temperature on thermal inactivation of E. coli under simulating minimal composting conditions suggested in the guidelines, 55 Celsius degrees for 3 consecutive days. I also tested whether E. coli VBNC cells could be induced under composting conditions and if they were able to resuscitate. At the end of heat treatment includes 3 consecutive days at 55 Celsius degrees. Heat-treated samples resulted in an inability to culture E. coli on agar plates. However, high concentration of live cells was still detected by live dead staining and PMA qPCR. The inability of E. coli cells to be cultured but remain live show that composting heat treatment induced cells into VBNC state. Amongst different types of resuscitation broth, bacterial waste from active growing cells performed the best for resuscitation of heat-treated cells. This suggested that compounds produced by active growing cells can trigger resuscitation. Here's the possible journey of E. coli onto fresh produce. Previous studies have suggested feedlot catcher manure can carry up to 1 million cells per gram of pathogenic E. coli. Based on the inactivation rate under composting conditions, after 3 consecutive days at 55 Celsius degrees, the population of culturable E. coli would decline to non-detected level, indicate die-off within microbial standards suggested by typical guidelines for finished compost, such as Australian standard. However, the risk is still there because E. coli cells have the potential to enter into VBNC states during composting. In the VBNC states, these cells go undetected by culture-based methods, thus providing a false impression of sanitation level. It is potential that growing bacteria may get introduced into finished composts after the heating treatment is over. Insects, wild animals, water runoff, and equipment used in compost handling may contaminate finished compost. Since composts are rich in nutrients, reintroduced pathogens may multiply under certain conditions. In addition, these actively growing cells may produce a stimuli that could resuscitate VBNC cells induced by the composting conditions. The regrowth of E. coli and Salmonella a long time after introduction of organic amendments into soil has been reported. This suggested that VBNC cells within the compost could be an important factor that facilitate the persistence of pathogen in manure-amended soils and thus pose a risk to microbial contamination on fresh produce. That's the end of my presentation. Thank you very much.
Thank you, Dao.